Good morning, guys. Welcome back to FECAM's unique histology review series. This video is brought to you by the academic and editorial boards of the Federation of Catholic Medical and Dental Students, FECAM's unique chapter. My name is Uguchukwu de Victor. Please do not forget to like our videos and subscribe to our channel, as this will encourage us to make more educational contents. Thank you. So today we'll be looking at the histology of the nervous tissue. So this is meant to be the last part of our histology review series. So the organs that we are going to focus on here includes the cerebrum, the spinal cord, the cerebellum, the peripheral nerve, and the myelinated nerve. So the cerebrum. So let's read the side note. The major reasons for identifying the cerebrum is the presence of pyramidal cells in the cortex. The development of the cerebrum is from the proencephalon, the telencephalon. The function of the cerebrum includes interpretation of impulse, vision, and it also controls emotion. The weight is about 1.2 kg, and then the pathologies involving the cerebrum includes encephalitis, uremic encephalopathy, cerebral palsy, multiple sclerosis, and stroke. So the structure of the cerebrum is divided into an outer gray matter and then an inner medulla. So covering the gray matter is the pia mater, which is the innermost layer of the meninges. So if you're looking at the histology of the cerebrum, the outer covering represents the pia mater, which is a layer of connective tissue. Now, in the gray matter, you see the neurons. The gray matter is the part of the cerebral cortex that contains the nerve cells, the cell bodies of the neurons. That's the reason why you see numerous black dots here, which signifies the nuclei, which represents the nuclei of the neurons. While if you go deeper, you'll notice that there's a deficiency of the cell nuclei, because you can't really see black dots inside here. So this tells you that this is the white matter, and the white matter contains the axons of the neurons. It mainly contains the axons of the neurons. So that's the white matter. Now, if you look at the cerebral cortex, you'll notice a kind of layering in the way these nerve cell bodies are distributed. So there are some areas that are faint staining, especially the outermost area that is faint staining. Looks like it contains fewer black dots or fewer neurons, contains fewer cell bodies, and then the deeper ones contains more of the black dots, and then there is a kind of gradual transition. So this represents the layers of the cerebral cortex. So let's look at other images. Same thing here. Look at the outer covering, which is the pia mater. Now look at the outermost part of the cortex, just deep to the pia mater. Notice how deficient it looks. Doesn't contain much of the black dots or the nuclei. Then look at the deeper layers. Notice that the dots increases, the nuclei increases. Same thing here. And then the pia mater. And then notice that the black dots increases. Same thing here. This kind of deepening of the outer cortex represents the gyri, the sulci, So the sulci is the deepening, and then this one is the gyri. So the sulci is a deepening. So the same thing that you see here. See this deepening here. This represents the sulci. Sulci is plural, sulcus is singular. So this represents the sulcus. Can you appreciate it? Now also notice that this part is deficient in nucleus, in nuclei. Doesn't contain many black dots. And then notice the gradual transition. Here contains numerous black dots. And then the gradual decrease. See this part too. This is the pia mater. Then this is the blood vessels within just deep to the pia mater, because it's a connective tissue, and connective tissue contains blood vessels. Now, just deep to the pia mater, you see the layer that is deficient in black dots. It contains a few black dots though, but then it's not numerous. Then this, notice the gradual increase in the cells, in the cell bodies. So those black dots that you are seeing represent the nuclei of the pyramidal cells in the cerebrum. So notice this again. This is the pia mater. Notice the blood vessels, these circular structures within the pia mater. Now notice the area just deep to the pia mater appears deficient in black dots. So this represents the outer molecular layer. So it contains majorly the cell processes. Cell processes. It contains a few scattered neurons and more of the cell processes. Now, the deeper layers, they contain more of the black dots. Now, same thing here, pia mater. Then you notice a layer deficient in neurons. So it contains a few neurons, but then it has more processes. Then as you go deeper, you see more black dots. So the staining here is silver impregnation or the Kajal method. So now let's list the layers of the cerebrum. So the outermost layer, just deep to the pia mater, is the molecular layer. So this is the molecular layer. Then the layer just deep to the molecular layer represents the external granular layer. And the external granular layer contains densely packed nuclei. So you see numerous, see how densely packed it looks. Can you appreciate it? How densely packed it looks. So let's look at this image here. So notice the internal granular, the external granular layer, just deep to the molecular layer. Notice how densely packed. If you zoom into this one, you notice it. You notice the molecular layer, it is deficient. Then notice the layer just deep to it, which is the external granular layer. So it contains densely packed nuclei. Can you appreciate it? Then the layer just deep to the external granular layer is the pyramidal cell layer. So here you find large triangular cells, which are the pyramidal cells. So you see large triangular cells. So this is a classic image, and you can appreciate the triangular pyramidal cells. Then the layer just deep to the external pyramidal cell layer is the internal granular layer. So the internal granular layer contains some stellate neurons. Then the layer just deep to this internal granular layer represents the internal pyramidal layer. So this is another layer of pyramidal cells, and you can notice how the pyramidal cells are shaped. So this layer contains large pale cells. Notice how large the cells here appear relative to other areas. Notice how large the cells are. Now, the deepest layer represents the multiform layer. So in the multiform layer, you find cells of varying sizes. So you see cells of varying sizes here. So the arrow here is pointed at pyramidal cells. So same thing here. Notice the pyramidal cell layer, which contains the large triangular cells. Then this is the external granular layer, which contains the dense packed nuclei. Then this is the molecular layer, which is deficient in cells. It contains a few scattered neurons and more of the cell processes. So, and so on. So same thing here. The deficient molecular layer, cell deficient molecular layer, contains a few cells and more processes. Then you see the more densely packed area. This is the cell deficient molecular layer. Then the densely packed area. Then you see the pyramidal cell layer, more of triangular neurons, large triangular neurons. So same thing here. This is the sulcus. The sulcus. So here the layers of the cerebrum is labeled. Then you can also appreciate the things we've mentioned before in the external granular layer, which contains dense packed cells. Then the pyramidal cell layer, and so on and so forth. So the slide of the cerebrum is very easy to identify. You can't mistake it for the pineal gland, because in the pineal gland, the dots here are scattered all around the slide. And then you can also find these large black irregular masses, which is called the pineal concretions. So you don't find something like this in the cerebrum. So you see the pineal concretions and then the numerous scattered cells all over the slide. So this doesn't look like the cerebrum. And then it doesn't look like the adrenal gland either. So this is the pineal gland again. You can see the pineal concretions and then the scattered cells. Same thing here. Then the suprarenal gland, you see a kind of sharp transition, and you see only 3 layers in the cortex. And then the medulla will contain numerous sinusoids. So these white, these red stuffs here represents the sinusoids. So you don't see something similar in the CNS, in the brain, in the cerebrum. So the slide of the cerebrum is easy to identify. Now the next one is the cerebellum. So the cerebellar cortex. The reasons for identifying the cerebellar cortex depends on the pointed parts. If no part is pointed, list at least 2 of these: A. Purkinje cells, molecular layer, or granular layer. Development is from the rhombic lip of the metencephalon. The function is coordination of growth. Is the coordination of movement and balance. Weight is about 150 gram. Pathology is cerebellar atrophy, cerebellar herniation. So for unique histology steeplechase exams, if no part is pointed, then you have to mention at least two of these: the Purkinje cell layer, molecular cell layer, or the granular layer. So this concerns your unique histology steeplechase exams. Okay. Now, when you are faced with the slide of the cerebellum, it's quite easy to identify in that it looks like trees. Look at these things, they look like trees. Let's look at other slides and then see this one too. But then notice one thing that is consistent. It looks like the branches of the tree contains a deeper staining structure inside it. Look at this. This one looks like tree also, with a deeper staining structure inside it. So this is just like a part of the cerebellum which is cut out. Okay, so for the cerebellum, the cortex is also layered, just like you see in the cerebrum. But the cortex here contains three major parts: the outer, molecular layer, then a single layer of pyramidal of Purkinje cells. So note here that you find Purkinje cells and not pyramidal cells. So the margin of this pale staining area and this deep staining area. So there is a layer of pyramidal Purkinje cells, and then this deep staining area represents the granular layer. So you have molecular layer, then a layer of Purkinje cells, a single layer, and then you have a inner granular layer, which is this deeper staining part. Then this thin pale staining area within the granular layer represents the white matter of the cerebellum. So from the granular layer outwards, that represents the gray matter of the cerebellum. Okay, now this is the cerebellum zoomed in. So this pale staining area, just like we've seen, this area that we've seen here, this area that we see here is the molecular layer. So the molecular layer contains a few cells as well, so it doesn't stain much. That's what gives histology images a thick staining structure is majorly due to the presence of nucleus. So the more it stains with H and E, the more you see darker structures, which represents the nuclei, the more the cells. So here you see a few dark nuclei. So this means that it contains a few cells. So most of the cells here are stellate cells and then basket cells. Now this is the single layer of Purkinje cells. Notice that they appear flask shaped and then they are evenly spaced. This is the layer of Purkinje cells. And then the granular layer, which contains densely packed nuclei, meaning that there are numerous cells here, cell bodies here. Notice the same arrangement here. You see less of nuclei here, which represents the molecular layer. So here contains more of cell processes Now look at the Purkinje cell layer, which contains flask-shaped cells. And then they are arranged in a single file. Then the granular layer, which contains densely packed cells. So the same arrangement here. This is the molecular layer, labeled as M. Then this is the single file. Let's zoom in. Can you appreciate the flask-shaped cells? This one, this one, this one, this one, this one, this one. So notice that they are uniformly spaced. So this represents the Purkinje cells. And then you can see the granular layer, which contains densely packed nuclei. So most of the cells here includes the Golgi cells for the granular layer. So the molecular layer contains stellate cells and then some basket cells. The Purkinje layer contains the Purkinje cells. So you can appreciate the same thing here, the same idea here. This is the outer molecular layer, or the molecular layer which doesn't take up stain. Well, because it has a few nuclei. Then this single layer is the layer of Purkinje cells. And now this deep staining area is the granular layer, which contains small ganglionic cells. Then the white matter, which doesn't stain well. So the same idea here. This is the white matter, the cerebellum, the granular layer, then the single layer of Purkinje cells at the margin, and then the molecular layer. So this looks like a tree, and then kind of looks like a tree. So same thing here. Looks like a tree with staining inside. So this slide is very easy to identify on histology. So that's it for cerebellum. So this is also silver staining by Kajal's method. Now the next one that we are going to look at is the spinal cord. So the spinal cord is classic. So you see a butterfly-shaped slide, a butterfly shape within the substance of the slide. So this butterfly shape represents the gray matter, and the gray matter contains the cell body of neurons. So the reasons for identifying this spinal cord includes the central canal, which is this space inside, the butterfly-shaped central gray matter. Now, multipolar neurons are found within the gray matter. So multipolar neurons are neurons with multiple processes. So they kind of appear like this. So you see the multiple processes and then the nuclei. So this is how a multipolar neuron is shaped. Now, so this is the gray matter, and then this is the white matter. Now this is the meninges of the spinal cord. So the white matter is this part. So this is the anterior part of the spinal cord. The anterior half is from here to here. So from here to here is the anterior. Then from this level down is the posterior. So this is the posterior gray column. This is the anterior gray column. And then this is the anterior median fissure. The posterior sulcus is not visible here. So this is the posterior white column. This is the anterior white column. This is the anterior white column. This is the lateral white column. This is the lateral white column. Now this is the lateral, this thing processes the lateral gray horn. The lateral gray horn or the lateral gray column. So this is the anterior gray column or the ventral horn. So this is the posterior gray column or the dorsal horn. So it looks like horn. So the function of the spinal cord is central nervous control of the body. The length is about 45 cm. The pathologies involving the spinal cord includes cord injury, cord compression, spinal cord concussion. So same idea here. Look at the butterfly shape. Now see the central canal, this white circle inside. Now this is the anterior commissure, anterior median commissure. This is the posterior sulcus. So you can appreciate the butterfly. See the butterfly? At the center of the butterfly, you see the central canal. And you can see the white column. And then you can see the meninges that covers it. So the same thing. Now, this is also the spinal cord. You can also appreciate the features here. So the stain used there is Luxor Fast Blue. So it's not H&E stain. Now the spinal cord, when you zoom into the gray matter, which is this, when you zoom in, this is what you are going to see. Multipolar neuron. I've drawn how it looks like, multiple processes. So this is classical for the spinal cord. The multipolar neuron has a clear zone around them on histology. So look at this neuron here with multiple processes. Notice the clear zone around it. Same thing here. Same thing here. So for all of them, you notice a clear zone around them. So that's the multipolar neurons. So same thing here. You can notice the clear zone around it. Then cells with numerous processes. More than one process. Process is just like an extension from the cell body. So more than one process means multipolar. So they are comparing the multipolar neurons of the spinal cord and the Purkinje fibers of the cerebellum. So notice the Purkinje cell layer. Notice the flask-shaped cells. Can you notice it? At the margin of the molecular layer and the granular layer. Granular layer is densely packed, and then Purkinje cell layer at the border of the molecular and granular layer of the cerebellum, and then the molecular layer. Now the peripheral nerve. The peripheral nerve is simple to identify because it appears black on most histology stains. So the peripheral nerve is structured in such a way that the single nerve fiber is surrounded by myelin sheath. So the stain used for this is called the osmic acid. So osmic acid stains myelin sheath black. It turns it to black color. So that's why you see these round rings. So the myelin sheath surrounds the nerve fibers, the axons. So you can see this. Then the nerve fibers are arranged in bundles or fascicles. So this is the fascicles. So the fascicles is lined by perineuron. Now, each single nerve fiber is surrounded by endoneuron. Then a bundle of nerve fibers is surrounded by perineuron. So the perineuron covers the fascicles. Now, if you bring numerous fascicles together, it will be bounded by an outer epineuron. So when you bring the numerous fascicles together, it will be bounded by the epineuron. So this a perineuron because it contains numerous axons. Then, so this single bundle is a fascicle. So if you bring this one, so the fascicle is lined by perineuron. If you bring 1, 2, 3, 4 and bind them together into a single nerve fiber, the covering will now be the epineuron. So that's how the layering is. So it kind of looks similar to the arrangement in muscles too. So this is perineuron. So the reasons for identifying the peripheral nerve includes presence of perineuron, presence of epineuron, and then presence of endoneuron. So the endoneuron it covers the single nerve cell, the single nerve axon. Now multiple axons bounded together forms the fascicles. Then numerous fascicles bounded together is covered by the epineuron. So this is the end of the histology review series. So I hope you've learned a lot from these videos. Now go over the slides again and you'll understand them. These are our sources of image. So these websites are great sites for learning histology. Contains numerous images. These two. This one too. This one too. This one too. So these are our sources. So this is where we are going to stop for this review series. Drop your questions and comments in the comment section. Thanks for watching.